My name is Adarsh, and today we'll be learning about transcription and translation. So, as we know that human body it consists of cells, and in cells we have different organelles present and different materials. So, looking into it, so we have nucleus and we have cytoplasm inside the cell. So, this as we know that cytoplasm consists of different materials, it has different things in it, different organelles as well. So we have, including those, we also have amino acid inside the cytoplasm. And we have 20 different kinds of amino acids. We'll get back to this. Now we'll look into nucleus. So nucleus, we have genetic material inside the nucleus, which is called chromosomes. So we have 46 chromosomes in our genetic material, or 23 pair, each comes from each parent. When we closely look at it, so we see that our genetic material, it's cleverly bound onto the proteins called histones. And when we unfold it, we see that it consists of a double helix structure, which is called DNA. It's a deoxyribonucleic acid, which is a double-stranded helix. Now, for DNA, we also have these genes on DNA which do different kind of things. It helps in the protein synthesis and enzyme synthesis and different things. So in order to make those proteins, we need this DNA to be converted into RNA. We'll get back to that. But why do we need our DNA to convert into something different so that it could cross the nucleus and go into the cytoplasm and make protein? Because DNA molecules, they're too big. They're too big to go or to travel from the nucleus into the cytoplasm through the nuclear pores. And also we don't want our DNA to be into the cytoplasm because we have lots of different enzymes there that could degrade the DNA. Therefore, what body does? Body converts our DNA, which is a double helix strand, into a single helix strand. By doing this, what is it causing? It's making our DNA into a single strand, or it is making it into a template that could act as a template for the messenger RNA synthesis. But how this happens? So DNA is converted from a double helix to a single with the help of enzyme called DNA helicase. So when this DNA helicase, what it does, it breaks the hydrogen bond between the bases of the DNA. It breaks the hydrogen bond, converts the DNA double helix into a single strand, which then acts as a template for messenger RNA synthesis. Now we have this template of DNA, and then we also have free-floating RNA nucleotides present in the nucleus. These RNA nucleotides will complementary pair with the bases of the DNA template. And how would it does that? These nucleotides will attach to the bases of the DNA with the help of RNA polymerase enzyme. This RNA polymerase enzyme will bind together or hold these RNA nucleotides complement to these bases and it makes the messenger RNA. So this whole process of DNA into mRNA conversion is called transcription. Now what we have here is the mRNA, but it's not the mRNA that would make the protein. It's the pre-mRNA. It's in a stage where it has different regions called introns and exons. So introns are the regions in the mRNA that don't code for the amino acids. Or these are the regions that don't code for the amino acids. So that's why we don't need these introns in our messenger RNA. We only need those regions that code for the amino acids so that it can help in the protein synthesis. So these are removed from the introns by a process called splicing. So these spliceosomes, they would break these introns from this pre-messenger RNA and would give us a messenger RNA with the cap end and a polydyl A with these 4 nucleotides, adenosine. Now after splicing, we get this mRNA that can now move from nucleus via nuclear pore and travel into the cytoplasm and assist in the making of proteins. Now, from there, we'll get back to this messenger RNA, that what it does and what happens to it when it goes into the cytoplasm. We need other kind of RNAs that would assist in the protein synthesis. We have transfer RNA, which is also prepared into the nucleus. Same DNA, double helix would then convert into a template. This template, through the process of transcription, this template would give us a pre-tRNA, again through the same process, and then the splicing will occur, which would remove the introns from the pre-tRNA, and after the removal we will only get the exons, and this is the tRNA which we need in the cytoplasm for protein synthesis. Once this tRNA moves from nucleus into the cytoplasm, it attains a shape like this. It conforms into a 3D structure or shape that's more like this. Now we know that tRNA, it's only made up of nucleotides, and these assist in the synthesis of protein. What it does, the tRNA attaches, one tRNA attaches to one single amino acid. So we discussed at the beginning that cytoplasm contains amino acids, and now this tRNA would attach to amino acid. But how would that happen? So we have this amino acyl tRNA synthetase enzyme that's present inside the cytoplasm. It would help in the attachment of tRNA to amino acid. So one tRNA would attach to a specific amino acid with the help of this enzyme, and then we'll get a structure like this. So we have tRNA and we have amino acid. And as we discussed earlier, that all the RNAs, they are made up of nucleotides. Similarly with tRNA, it's also made up of nucleotides. So we have these nucleotides, and these three nucleotides are very important, detached at the bottom for the protein synthesis. Now we have two main things that are needed for protein synthesis. We have tRNA and we have mRNA. Now we need ribosome, which is called the protein factory. So what happens if we go back into the nucleus and dig a bit deeper, and we go into the nucleus, we have DNA which converts into pre-RNA through transcription, and then splicing occurs, and then we get two RNA subunits. Instead of getting one pre-mRNA as we got here, or one tRNA, we get two subunits, one large subunit of RNA, and one small subunit of ribosomal RNA. Once we get this, these would then leave the nucleus, go from the nuclear pores into the cytoplasm. Now what happens, these ribosomes, they attach to the messenger RNA, the larger subunit of the cytoplasm, ribosome, and the smaller subunit of the ribosome. They attach to the messenger RNA. So we get this ribosomal unit. So we have this messenger RNA, now we have this ribosome. It attaches to the messenger RNAs and start to travel from the cap end towards the polytail A end. Meanwhile, when it's moving, what it is also doing is that this tRNA, which was already in the cytoplasm with an amino acid attached, this ribosome causes this tRNA nucleotides to match with the nucleotides of the messenger RNA. When this, it's called anticodon on tRNA, and on messenger codon, these three are called codons. When this codon attaches with this, matches with this anticodon, the ribosome helps to attach this tRNA to the messenger RNA. It helps assist in the attachment. And then what it causes the tRNA to move forward, and then releases the amino acid, and after releasing the amino acid, the tRNA would exit the ribosome, and the ribosomal units would still be moving towards the polytail A, and during this process it would match, release amino acid, and exit the ribosome. And this released amino acid will attach with the other amino acid from other tRNAs and would make a protein. So this whole process is called translation. So the ribosomes are helping messenger RNA or translating messenger RNA to make proteins. So now what we studied in all of this, so we have transcription and translation. Transcription means that we have our DNA, our genetic material, that converts into a single strand, act as a template for the preparation or for the making of pre-messenger RNA. Then through splicing, it makes a messenger RNA. Making of this mRNA is called transcription. Again, we have ribosomal RNA and tRNA made into this from our genetic material via a process called transcription. Then these go into the cytoplasm, and then we have this ribosomal subunit that act as a protein factory and assist in messenger RNA and tRNA for the protein synthesis, and this whole process is called translation. And just for your knowledge, once we get this protein from the messenger RNA, this can be broken down again into amino acid, and the whole process could continue. So that's it for today.